From Microbe TV, this is TWiEVO, This Week in Evolution, Episode 20, recorded on June 12th, 2017. This episode of TWiEVO is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get your first 3 meals free with your first purchase and free shipping by going to blueapron.com/twee. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels, Elde.
Hey, Vincent. Good to be with you again.
Seems like a long time, but it wasn't that much, right?
Yeah, not too long. A couple weeks ago, we got back in the saddle and talked to Jonathan Weiner. A really fun conversation to hear about his science writing, his career in general.
Yeah, and since then, I finished up Time, Love, Memory. You read that
You and me
one,
both.
Right?
Yep, exactly. Yep, it's a thick book, lots to read, very dense and rich.
Yeah, I had
really fun.
I had a paperback copy with tiny print. Oh my gosh, and when I traveled, I was reading the digital thing where you could blow it up as big as you want. Yep. That's a great book. Just to learn about the history of genetics and molecular biology and the people who did it. Wow, it's just amazing.
Yep, definitely right up there near the top on our TWiEVO reading list, which we need to put together.
And today I got another great book for our pick later on, something I learned about on my travels.
Oh, cool. I look forward to hearing that.
We'll do that.
Speaking of travel, it sounds like you've been on the road.
Well, I was at ASM Microbe in New Orleans a week ago. It was over the last weekend. And we did a TWiV, which is out with astronaut Kate Rubins. So that's pretty cool. You should check that out. And then we also did a TWiM there as well. That's not out yet. That'll be out. What is today? Monday, this coming Thursday. That was with Arturo Casadeval.
Oh, very good.
Talked a lot.
Must have been a fun conversation.
Oh, so he's written like a number of position papers on what's wrong with science and how to fix it. So we talked about those mainly, and that was a lot of fun. I also did a bunch of interviews with people, and so they're all there on the ASM website. I'll put links to them. And then, of course, in 2 weeks, we have the American Society for Virology.
Yeah, I'll be there up in Madison, Wisconsin.
Yeah. Not too far for you to go, right?
Not too far. Maybe to put in a quick pitch. So I will be there as part of the Saturday set of satellite symposia. So Colin Parrish, who's at Cornell University, is organizing one through the Ecology and Evolution Club. And the topic is virus and host evolutionary co-selection and arms races. Host interactions and coevolution among viruses. So I'm really looking forward to it. I'll be the first speaker, and then there's a lineup going through the day of some great virologists who think about evolution. Yep,
I registered for that. I'll be there.
Cool.
Yeah, that's those Saturdays are good because they're small. They're like 100, 150 people, and it's before all the crowds descend.
Yeah. Yep, kind of good chance to have conversations, eat lunch together, kind of compare notes.
So it was 2 years ago, Nels, at an ASV that you and I talked about doing TWiEVO, right?
That's right, up in London, Ontario, Canada. That's right.
That's right. And here we are at episode 20, big milestone.
That's right, 20. Who'd have thought?
Well, maybe not I guess so big, but
it is for me. And I don't know how you've done much more podcasting than me, but I guess in podcast years, we're now near the legal age of drinking as we approach 21.
Well, we've gotten past
episodes.
We certainly have gotten past the number where most podcasts fail at. Right?
Wow, that's a good sign. I have to say I'm having a ton of fun. So I feel like we're just gaining steam, if anything.
Yeah, well, we got a number of fans. People like it, as we'll see in the emails. And, you just have to keep at it, and you get more and more people listening. So you should try different things. We should experiment as well.
Yeah, I was just thinking about that with this sort of half-baked drinking age idea. Have you ever seen the show Drunk History with, I forget the person who runs it, Derek Waters or something like that?
No, I've heard of it, though.
Yeah, so what he does is he brings on, usually a historian to tell a story about the historical event or time period that they're interested in. Usually they pick a pretty focused story. But the sort of twist on it is they show up completely inebriated. Things go things go off the rails pretty quickly. And then they actually have, as the person is telling the story in sort of a sloppy way, they actually show actors who are playing out the scenes as if it's happening and voicing over the story told by the historian. It's really hilarious, actually. Might be a little too much for TWiEVO at this stage, but I'm definitely up for some experiments from time to time.
There was a podcast, forgot the name of it, many years ago at the beginning of podcast history. They would drink beer during the show.
Oh, interesting. Kind of promote a casual conversation in a sense.
Yeah, it would loosen them up and
sure,
get a little crazy. Yeah, I forgot the name of that one. Yeah.
Interesting.
We were waiting for TWiM to start in New Orleans. I said to our tour was Ray Ortega was out there in the audience, and he said, "Need anything?" I said, "Yeah, a glass of red wine would be nice."
There you go. Yeah, I was sad to miss the ASM meeting this year. My postdoctoral mentor, Harmeet Mullik, got a really nice award. And then had some friends out there as well. So I was just trying to follow it from Twitter, and a lot of people who were, including you, who were putting up really nice summaries of some of the events there. And so at least it felt like you could follow along.
Yeah, it's a cool thing. Enough people now are tweeting. You know, Nels, I joined Twitter a long time ago.
Right. Yeah.
2008. And there were no scientists around, you know.
I know.
There was nothing from meetings. Now you can get all kinds of information. Your old buddy Cedric Fichat, right? Yep. I'm following him, and I'm watching him move to Cornell.
Yeah, exactly. We can see his campsites every evening.
And he's always tweeting good stuff about science. And also a bunch of other people are there. It's totally different now. It's great.
Agree. Yeah, Twitter is...
Even Hopi is on Twitter, and she's always tweeting about stuff. It's really great.
That's right. It's really fun. I think Twitter is kind of tailor-made for science conversations somehow. It seems like that Twitter itself, just in general, is probably not doing as well as at least the shareholders were hoping.
Yeah,
if you look at the stock price, it's kind of slipping. But I think it's only getting more and more vibrant for these kind of scientific exchanges of information, which is really, really great.
Yeah, and certainly new thing, because when I started, there was nothing like it. I was following a couple of science writers and so forth, and only now in the last, I would say, 2 years has it really come into its own for science, and that's great.
Yep, really fun.
Yep. Well, let's move on to our work here on TWiEVO.
Yeah, good idea. So we've had some really fun interviews the last several episodes, and so it seems like a good idea to get back in the trenches a little bit with some new work, and and maybe have you and me kick it around a little bit,
sure.
And see where we end up. So I've been thinking for a while that it would be really fun to grab a paper when an interesting one emerged, thinking about symbiosis and some of these more mutualistic interactions or associations between different species. And, I think both of us with sort of virology or microbiology backgrounds or interests in that, sometimes a lot of the work we focus on can have more sort of clear antagonistic sort of transactions between species. But there's burgeoning and growing, of course, massive fields of really interesting work, thinking about sort of the other side maybe of the same coin, which is how these associations between different critters can actually be at least temporarily beneficial, and even in sort of long-term scenarios and associations.
Yeah, we talk about symbiosis a lot over on TWiM. And one of the trends we've noticed, we did a paper a couple of weeks ago, maybe a week ago, on symbiosis between an algae and a salamander.
Yeah, that was a cool one. I saw that. Yeah. And so is that episode out yet?
It came out. It came out last week. The cool thing now is that what people are doing, and as we'll see, this applies to today as well. You can look in both members of this symbiosis and look at how gene expression changes and get clues about how it works. You could delineate some genes that go up or down, and then say, okay, let's artificially alter those and see if it affects the symbiosis. It's really amazing what you can do.
Yeah, I agree. It feels like we're kind of just in this, new, again, kind of stretching back to what we've been talking about on TWiEVO, sort of this new era of possibilities given the genomics and sort of as people are examining these diverse systems involving salamanders, or we'll talk about cnidarians today, it just kind of opens these new doors, which are really exciting to think about.
Now, over on TWiM, we said last time, well, the Earth is one big symbiosis, really.
There you go.
I don't know, maybe at some point in your education, you had this view that everything is living pretty much on its own, and occasionally, species eat each other.
Right.
But I don't think we appreciated the extent to which different species live on in each other in pretty beneficial arrangements, right?
Yeah, that's right. I mean, I would definitely say just, even within the last 5-10 years, kind of the realization of the importance of the microbiome in our own guts has started to illustrate this to a broader audience. I think one of the really cool kind of twists on some of these symbiotic stories, and something we'll talk about a bit today, is how there's actually even though it might not be an obvious antagonistic interaction, there are these sort of immune-related transactions that occur when something that is either, a very transient or a symbiosis or an association which happens in every generation. These species have to delineate whether something foreign and possibly sinister is coming into their, neighborhood, or literally into the organism itself, or if this is something beneficial. And so how that kind of molecular conversation, as it's, I think, interestingly described in this paper, it becomes a really cool window into how these things are working.
So there's some symbioses that get reestablished continuously, right? So there's the squid, the bobtail squid with its bacteria. Mm-hmm. They, at night, the bacteria provide light, so the squid appear invisible in the water from below.
Yeah, isn't that
they're
cool?
Not predated. Yeah. And then when in the morning, the squid, they go in the sand and the bacteria go away, and then they recolonize just before it dies. So you have to, and there are many examples of that where they come and go, the symbionts. Mm-hmm. So you have to figure out how they can do that properly. But then there are long-term symbioses like ours, our gut microbiome for one. Mm-hmm. But still, you have to make sure we don't kick them out, right?
Yeah, absolutely.
And we have to make sure they don't get pathogenic, which can happen if you take antibiotics, for example.
That's right, too. Yep. Or I would say even, from those very transient associations you're describing in the bobtail squid with Vibrio, or what appear to be very permanent associations. So the mitochondria in each of our cells, for example, or most of our, the vast majority of our cells, which have been hanging out there for, a billion plus years. And so it's the whole spectrum of transient and semi or completely permanent interactions as these formerly independent species start to depend on each other or become obligate or rely on each other. And you kind of can't go back at some point.
Yep. Yeah, so you picked up this paper from eLife, huh?
Yeah, that's right. So this paper kind of came across my radar screen a few weeks ago. I think someone tweeted about it as a sort of what's happening these days. And so it's the mutualism that's being investigated is one between cnidarians. So these are mostly, so certainly aquatic, mostly seawater, with some freshwater exceptions and some interesting freshwater exceptions, actually. But so some of the common cnidarians, of course, are sea anemones and corals and sea pens and so forth, even jellyfish and other species like that. And then the association are with dinoflagellates. And so these are the ones that the authors consider for this work associate with, these are dinoflagellates that associate with cnidarians. These are algae, and they're providing key nutrients as they associate with the cnidarians. Things like corals, for example, are some really well studied examples, and then getting at that point of how long have these been associated. A lot of these are acquired each generation, so it's not quite as transient or volatile as that association between the bobtail squid and their bacteria. But this is happening with reproduction, and in some cases these are horizontally transferred in the sense that with each generation that association has to be made de novo. And then there are other species that have been described for vertical transmission, meaning that the sort of mother, so to speak, is passing this on to the offspring. And so that's a slightly more stable route of transmission.
I should point out that cnidarian has a C in front of the N, which is silent.
Mm-hmm.
So if you have trouble reading the paper there. Nels, are there any cnidarians in Utah?
Great question. I would say my suspicion would be yes. And the reason I'm saying that is I'm forgetting the details at the moment. But I remember seeing it was a paper or maybe even an article recently that someone discovered a parasite of the trout up in Idaho, state bordering us to the north. And it was sort of a weird, the critter wasn't so clear what it was. And then I believe they sequenced its genome. And in fact, what they had on their hands was a freshwater cnidarian in the Mountain West and in freshwater. And so I wouldn't be surprised if we're in the company of cnidarians.
So most of them are marine animals, but there are some freshwaters. Yeah.
Yeah, indeed. Yep. And some really weird ones that we're just, kind of glimpsing at for the first time, or even just, discovering that they're hanging out kind of under our noses in puddles and ponds and streams.
So this is an eLife paper, so it's open access.
Open access, which is good news. And one of the things I like about these eLife papers, and we might get into this a bit later, is, you could actually get a glimpse behind the curtain of the review process where they post a summary of what the reviewer's comments were and how the authors addressed them.
Yeah.
And so the title of our paper today is a diverse host Thrombus Bondin type 1 repeat protein repertoire promotes symbiont colonization during establishment of cnidarian dinoflagellate symbiosis.
Not exactly a New York Times headline.
Yeah, it's a little bit wordy. It's a dense title. But it gets at, so what we'll be focusing on in our conversation is this interesting family of proteins, these so-called Thrombus Bondins. And we'll talk a bit about that. We've already kind of hinted at this cnidarian dinoflagellate association, these algae that associate with, and in particular, the species they'll be looking at are marine cnidarians, both sea anemones and I think corals. I think it might be all anemones. And the authors of the paper, this is Emily Fleur-Neumbauer, Angela Poole, Philip Neumbauer, Olivier de Tournay, Kenneth Tan, Simon Davey, and Virginia Weiss. And so the two senior authors, Virginia Weiss, she's at Oregon State University in the Integrative Biology Department. Simon Davey is in New Zealand at the Victoria University of Wellington. And then some of their associates on this paper.
There are two Neubauers. I wonder if they're related in any way.
Well, that's a good question. It seems suspicious.
Well, it could be a number of possibilities. Could be siblings, could be coincidental, or they could be partners.
That's right.
Is there anything I leave out? I want to please everyone,
I think that covers the ground. If anyone who knows either of these authors is listening, maybe you could tip us off.
Do you know any of these individuals?
I don't think I've met anyone on this list. Yeah. Been to University of Oregon somewhat recently. They're doing, there's a great program run by Karen Gilliman on mostly microbiome, but some associations between species as well, kind of a systems biology approach. And so there's some really great work going on in that neighborhood, including this interesting work that I didn't realize was happening there.
This protein is in the headline. It's very, very prominent. Thrombospondin type 1 repeat protein. Do you know anything about that? Have you encountered those before, Nels?
I think I haven't taken a deep dive on any of these. I've heard of them before. One of the things I have to say, it's really interesting reading the introduction of this paper, is they talk about sort of the probably best described biology of the thrombospondins. And so just a little bit of background. These are repeat proteins, so they have these motifs. They go into some detail to document sort of the sets of factors that you see. So these so-called TSR domains or thrombospondin domains, which are common not only in sort of these sea critters, cnidarians, but also all the way out to humans. And that's where most of the functional work has been done to date. And in some cases, actually, in terms of immunology and tolerance. So these, interactions between different tissues or cells, recognizing self from non-self and making sure that you don't turn on an immune response that would, kill yourself, basically, in an autoimmune scenario.
And that makes sense in terms of a symbiosis, right?
Yeah, exactly. Isn't that interesting? So I kind of love this idea that it's almost like reverse model systems genetics, where you take what's known about human biology, and then you kind of recast that to think about these, cnidarians or these symbioses. And so it's like all of the NIH funded work, in a sense, is now reflecting back out to the field into these sort of, ecological interactions.
Yeah.
And then I think the best case scenario is it becomes almost like a two-way conversation where what we've learned about the human immune system or tolerance or things like this might inform what we learn about symbioses, which in turn might inform what we understand about our own biology. And so I really enjoy these kind of,
for sure.
Yeah. Yep. It's neat interest.
There's also some role of these TSR proteins in attachment, right?
Yeah, that's right. And so a lot of these are receptors. They're membrane associated. They have signal sequences, so they get out to the membrane where they can interact with other proteins. And in fact, one of the things, and I didn't realize just how diverse this family of proteins are. So in figure 2 of the paper, for example, they just show some of the places where you see these Thrombus Bondin repeat domains showing up. And it's not just, they're not just there alone. So you see these things actually in some old friends like trypsin, associated with some of those domains, von Willebrand factor, which is a well-studied protein involved in regulated exocytosis. They end up hitching rides in open reading frames with immunoglobulin domains, metalloproteases, semaphorins, which we'll dip back into in a little while. And so these things are just really kind of distributed or dispersed through genomes in really interesting ways.
Yeah, it's with a wide range of functions too, right?
Yeah, indeed. And so it's kind of interesting, but it also, I think, raises an important challenge, which the paper certainly kind of, or the work sort of finds itself in the middle of, is how do you sort of distinguish what is just diversification or the complexity of these protein families to actually sort of pin down or begin to get a sense for the functions that these things might actually be mediating when it comes to, for example, a symbiotic relationship in that conversation between two species.
Now, this isn't a random protein that they just said, let's study. There was already some evidence that it had something to do with functions in these cnidarians, right?
Yeah, exactly. And that's a really good point, because if you just read the abstract of the paper, it kind of comes out of left field where all of a sudden we're talking about Thrombus Bondins. And so that wasn't just sort of, opening a phone book and putting your finger down on one entry and saying, okay, let's see what happens. You're exactly right, Vincent. So there's been some work leading up to this paper, of course, which is how these things usually go, suggesting, in fact, that some of the Thrombus Bondins that you see in these sea critters that are symbionts might actually have some role. But the authors, and I don't know the field very well, but the authors state, and I think accurately, that a sort of functional foothold on this, or a molecular understanding of which ones are contributing to these interactions, is still an open question. And so that's kind of where they pick up with this work. But you're right. They had that very strong clue that this would be a good place to look if they wanted to start to understand the colonization events that happen when these dinoflagellates associate with the cnidarians.
Now, I have a question in terms of terminology. So the cnidarian dinoflagellate associated, that's a symbiosis, right?
Yeah.
And I would call both participants symbionts, but here they often refer to the host as one of the, which would be, of course, the cnidaria, and then the dinoflagellates get taken up into them. So they do refer to them as symbionts, and then sometimes to the host. What's your view of that?
Yeah, it's a good. I'm glad you're bringing up that point. I think it's interesting how I think we almost. I don't know if it's almost like philosophically we sort of have sympathy for the host and kind of think of it as the as the boss of the interaction. So the cnidaria, the reason this is considered a mutualistic or symbiotic interaction is that both sides really get something out of it, right? And so for the cnidaria, the idea is that when they associate with these algae and are exposed to sunlight, these things can undergo photosynthesis and then donate all of this energy to the cnidarian host, whether that's an anemone or a coral or whatever it is. Mm-hmm. And then on the dinoflagellate side, for the algae, the idea would be that by associating with this cnidarian, that this sort of offers a home or a protective, place to hang out, live. And so there's that would be sort of the beneficial scenario on both sides. To be honest with you, I kind of and I might be a bit of an outlier on my thinking here, but I kind of always take a sort of a sinister view of these associations, even though it seems like both sides are are getting something positive, which is true. My view on this is that when you collide two separate genomes under most circumstances, there's going to be some conflict some way or another at some point. It kind of builds out of the selfish gene idea. But certainly for the short term, you can see how this is beneficial. But, you could imagine at some point down the road, if all of a sudden a mutation arose in the dinoflagellate population, this thing swept through, and it could have a detrimental cost on the cnidarian host in some ways. It could crash both populations, for example. But, at the same time, it's very useful conceptually, I think, and certainly for these investigations to think about how there is a beneficial outcome on both sides. And I will, sort of put it out there that I don't even trust my own mitochondria. And just, who knows? There's a separate genome. At any time, it could turn on me and get in trouble.
It's a good point. In fact, this algae salamander symbiosis we talked about on TWiEVO, it's not even clear that what each side gets from this, right? It's an assumption that the algae goes inside the cell, the developing embryo, but they don't really know what is provided. And in fact, when they looked at the transcriptome of each, the host, a lot of defense proteins go up when the algae comes in, and the algae gets stressed out when it goes inside the salamander cells.
That's right. Yep. It's sort of like both sides are kind of, suspicious of the other one in terms of the molecular response there.
Yeah, as they should be, right?
There you go. Exactly.
So just because two organisms live really intimately, even with one within the other, doesn't mean that it's a symbiosis, right? You got to really prove it.
Yeah, I think. Well, it's an interesting point, and I have to confess that symbiotic relationships aren't really my field, and so I don't know where the state of the art is on that, on kind of those definitions or those conversations. But at least for me, it all feels like this continuum, and one that's dynamic, right? In the sense where something that's sort of beneficial on both sides today could be a disaster on one side or the other tomorrow, from an evolutionary standpoint.
Things could change. But I think that's a good point. It's a continuum. It's not black or white, right? It's not digital, not 0 or 1. There's just all different levels. And you have to just see what's going on. And as you said, I'd love the idea that it could change. Yeah.
Yeah. Yep. So and just maybe a little more background. So we've been talking a little bit about the bobtail squid, and I just thought I'd kind of highlight the work of a few labs in case any of our listeners kind of want to dig deeper on the topic. So, one of the labs that's done a ton of work, and other labs have emerged from hers, is Margaret McFall Nye's lab. And I think you did. Is it right that you guys did a TWiEVO episode on some of the work?
Yeah, we did. TWiEVO number 10. Long time ago.
Wow.
TWiEVO is up to 154 now.
Wow.
Margaret McFall Nye talked about the bobtail squid Vibrio symbiosis on that episode. She was actually a co-host on TWiEVO for a while.
Very cool.
In the very beginning, and that was sort of in the middle of her tenure. And then she went on a sabbatical and decided that she didn't want to do podcast anymore, so she's gone.
Yeah.
But
Okay.
TWiEVO number 10.
Cool. Yeah, and she also moved her lab at some point she was at UW-Madison for a number of years, and then now I think she's at the University of Hawaii.
Yeah, well, in fact, she was there at Hawaii originally. She moved to Madison.
Okay.
And she went back.
Yeah, there you go.
These squids, their native habitat is not Madison.
That's true. Exactly.
I think that was a big pull to bring her back to Hawaii.
Yeah. And then there's another whole kind of separate body of work that's come from Nancy Moran's lab. She's moved around a bit, but was at Arizona, then at Yale, now at UT Austin. And they've looked at a lot of I'd say in general, more kind of permanent symbiotic relationships between insects and bacteria. So things like aphids and cicada bugs. What's really fascinating about some of these systems, I would say, is that you have the whole range of kind of new associations between bacteria and insects, where it might be that they're just at the beginning of that sort of, consummating a symbiotic relationship, so to speak. And then ones that have been in association for a really long time, and some really cool work on how sort of genomic complexity emerges from some of those scenarios. And so some folks that were in Nancy's lab, I'm thinking of John McCutcheon, he's up at Montana, Colin Dale, who's actually here in my backyard in the biology department at the University of Utah, have picked up on some of those projects. Mm-hmm. So, and then there's even, I think thinking about TWiEVO and how people are starting to change fields or starting to add some more of this kind of evolutionary thinking or kind of going back to the diversity of life. John Pringle, who's now at Stanford, he was a yeast guy kind of coming up in his career and did some of the seminal cell cycle work with Lee Hartwell that led to the Nobel Prize some years ago. And he now has not completely, but almost switched his lab entirely to looking at model cnidarian and dinoflagellate symbiosis, some of the species that we'll be talking about today, and they've done some of the genomics along those lines.
Neat.
Yeah. And then the authors actually highlight a review. This is from Bob Goldstein and Nicole King. Nicole was on TWiEVO not too long ago. Yeah. And so they have I haven't had a chance to really dig into this, but I'd recommend it again to our listeners. So in Trends in Cell Biology, they have a recent review called The Future of Cell Biology, Emerging Model Systems. And they talk about some of the cnidarians as kind of cool emerging systems. So this is in Trends in Cell Biology. It's actually paywalled. But in any case, like not only this paper, but others that are not open access, like the eLife paper we're talking about, you can always email the authors directly. And in a case like this, just tell them that TWiEVO sent you. And I'm positive that they would be delighted to send reprints by email.
And we can get another TWiEVO listener.
There you go.
Goes like, what the hell is TWiEVO? And they'll say, oh, you should go check it out, microbe.tv/TWiEVO.
There you go.
Very cool.
So why don't we dig in here?
Yep.
Vincent. So we kind of set it up, this idea of these Thrombus Bondins, which might be proteins. So these are, expressed by the cnidarians, and so to begin with, so they kind of were focusing on that. But one of the reasons they thought that this might be a good place to look was that there are some data sets. So these are these high throughput transcriptome data sets, other omics approaches that were beginning to compare basically species that have symbionts, the dinoflagellate symbionts, and ones that don't, and whether you see, especially at the time when these, if the species come together, the dinoflagellates and the cnidarians come together, what genes are turned up with the idea that these could be candidates, in a sense, for ones that are important for helping to mediate that molecular conversation or those interactions between the two species.
So what's the name when they grow them without the symbionts? Asymbiotic, is that right?
Yes, or aposymbiotic, I think.
That's it.
APO. Yeah, APO. And so I'm glad you're bringing that up. I think it's an important point. So those are, from my reading, those are species that are usually symbionts, but they're in the state that they're being worked with or observed, they lack their symbionts. They're aposymbiotic. And that's an important distinction from species that are just non-symbionts, that they don't associate with these, these algae cells. And so in that case, if you're a cnidarian that doesn't have a symbiont, so how do they gain their nutrients? They're predators. They go out and feed on things like algae, but they digest them and gain energy that way. And so this is that sort of slight trick of, instead of treating these things like prey, is to just give them a home and let them create the energy for you. That's sort of the idea of the advantage there.
Yeah, they have a procedure for propagating them in the absence of the other symbiont, aposymbiotic anemone production there. So they have to grow them without the dinoflagellates, obviously. And they treat them in a certain way so that if there are any in them, they're going to get flushed out, basically, I think.
Right. Yeah, exactly.
I don't remember where that, it was funny that the way they said it in the experimental methods, we flushed out any potential dinoflagellates that were present here. So they basically get pure cultures of either one, then they can mix them together, as we'll see. Yeah,
exactly. And I agree. And I think having it also illustrates how having some ability to manipulate these things experimentally is really key to kind of moving forward, just from a kind of sequence gazing or comparing the family of proteins that you find in the genome to kind of move to the next level and start to test whether some of these might have an outcome or be involved in establishing these symbiotic relationships.
Yeah, they can grow both sides of this symbiosis in culture relatively easily. In fact, you can buy these cnidarians at pet stores. And I think they say that's the source of theirs. They originally got them at a pet store, and then of course they bring them in the lab and propagate them. Yeah, it's actually pet store is in the paper. I'm trying to find the exact citation. Here we go. A population of Symbiodinium minutum containing Apellida, so that is pallida, the two together, originating from a local pet store. Here's the whole thing, was maintained in a saltwater aquaria. Animals were rendered aposymbiotic by incubation for 8 hours at 4 degrees twice weekly for 6 weeks. So that's cold. They're giving it a cold shock. Followed by maintenance in the dark for 1 month. Anemones were fed twice weekly with brine shrimp and cleaned of expelled symbionts and food debris regularly. I think that's so funny.
That's great.
Expelled symbionts.
Yeah, kind of a high maintenance model organism here compared to, say, E. coli or baker's yeast, right? It takes a little more work to keep these things going, but that's what you have to do if you want to go after some of this unique biology, right?
Yeah, well, it's like any other system. Ants, right? You have to do special things, and
that's right.
So yeah. And the rewards are there for those who are willing to invest in the time, right?
That's right. I think you have to really, be passionate about your organism to invest in the what it takes to keep them going. But absolutely, that's the way to do it. Yeah.
So you said they had already some genome sequences of cnidarians. Is that right?
Yeah, that's right. And even better, they had some combination of data sets. And it's it reads a little bit like a mix and match of taking different data sets, kind of gazing at them, making comparisons. And so some of the data sets were from some of the symbiotic species. Others were from non-symbionts. And so you can make a case for presence and absence of a certain, family member of the Thrombus Bondin protein family, and maybe that could push you in the right direction. There's also these data sets and I think in the case of this paper, that were even more useful, were exactly what you just described, Vincent, with the setup. So they got some of these symbiotic cnidarians together, grew them for a long time, and then had others that were in association with their symbionts, with the algae. And then from there, it's kind of interesting. They went ahead and took a human antibody to Thrombus Bondin 1, I believe, and just asked if we look at the proteins being expressed. So grind up some cnidarians, right? And ask, what are the proteins being expressed? Do we see a difference by Western blot using this human antibody to that family? And so what you see then I think this is figure 4, is sort of the first result where they claim and show that there's sort of an extra band, or certainly a more prominent band, a protein band, in the symbiont case versus the apo symbiotic case. Yep. And so the argument.
Go ahead.
Yeah, go ahead. Well, I guess the argument would be, right, that there's this protein that's something like 47 kilodaltons, and is presumably in that family. And in fact, they haven't identified it, but they just see it by its sort of an increased presence, that it could be involved in these interactions.
Right. So they also do some immunohistochemistry on the tissues, right? And they can see it when there's a close association with resident dinoflagellates. Then they see the TSR proteins.
Yeah, exactly. And so if you kind of look at the immunofluorescence, there's, I think the red channel mostly is actually that's the dinoflagellates, and it's just autofluorescence in that channel. And then they use different channels or different wavelengths to see the localization in green of the at least what they presume to be a thrombus bondin protein based on its cross-reactivity with the antibody that was raised to a human thrombus bondin protein. And then they actually use the antibody, I think, in kind of a clever way, which is to not only look at fixed samples, but to actually add it to live samples.
Yeah.
In the apo case, right? And then see if and the idea is just by the protein interaction, the antibody binding to some set or a single thrombus bondin like gene or protein, that could potentially block the ability of these dinoflagellates, the algae, to associate with the cells. And that's sort of the first kind of functional evidence that they present that there could really be something going on here functionally with these.
So there's this experiment, they're actually taking the aposymbiotic anemones, which they've cured of dinoflagellates, and then they're adding dinoflagellates, right? Which are then going to get taken up into the anemone and colonizing, they call it. And so what does the antibody to TSR do to that?
Good question. And I don't think they cast a lot of light in that area, but what they do show is a couple of controls. So they either use the thrombus bondin antibody, like you're describing, an IgG control, or the FSW control. It took me a few minutes to find what that abbreviation stands for. It's filtered seawater, which would probably just be sort of business as usual, right? No extra protein hanging out. And what they show pretty clearly is the percent of colonization, that is the dinoflagellates infiltrating or becoming associated with the cnidarian, goes way down in the presence of the thrombus bondin antibody, but not the other two treatments. Yeah.
Amazing. It's really, I would never have predicted that or even done that experiment. It just seems too straightforward, right?
Yeah, kind of bold, it seems like. Yep.
So TSR proteins seem to be involved in the uptake, right? Yeah,
at least by that kind of that first experiment. They went on to do some kind of corollary experiments as well. And just to say, it's interesting when they're showing these graphs, basically over time in hours, how much colonization they get. And we should just say, so they measure that colonization. They take advantage of that autofluorescence again of the algae. And so what they do is after the treatment or just mixing the two species together, they just wait and see whether there's autofluorescence within the cnidarian as time continues. And then if it passes a certain threshold for, I think it's, the number of tentacles or the amount of autofluorescence you see, then they say, it's been colonized. And in fact, it's not like 100% of these things are getting colonized, right? It's only
it's not
15.
Much,
15%, but 15% compared to 0 or 1% is a pretty dramatic difference, I would say.
Yep.
Yeah, and then from there they get a little, they don't get super precise, and in the end, as we'll see, they haven't really nailed down the exact thrombus bondin containing protein that they're sort of envisioning as mediating this interaction, or even the mechanism of how this interaction might happen. But what they do is some variants on this where they have what they call peptide 1 and 2. So do you remember what these peptides were derived from? I believe it's from the they're at least getting a little bit closer. These are from the cnidarians themselves.
Yeah, they're short peptides they made based on the TSR sequence, right?
Exactly.
Conserved sequence. So it's a part of it,
Yeah, exactly. That they in that case they found it from those genomes, right? When they're characterizing the protein families. And so what they show is that you can actually, depending on the peptide you use, you can actually get so peptide 2, I think, was pretty potent for actually going a little above and beyond if you just have them in filtered seawater in terms of the amount of colonization. And so they make the case that this actually could be promoting the symbiotic interaction.
Yeah, they call it super colonization, right?
Yeah, exactly. And so I think that's a generous maybe designation, since you go from, let's say, 15% colonized up to 20%. That's super. It's certainly increased, and they do some careful statistics, some ANOVA testing to try to bolster the case. But so that does, I mean, what's nice about that, right, is then you kind of see that even though it's not completely specific, you see the effect in both directions, which is if you block it with that antibody, you can take it down, or if you add this peptide, the thrombus spondin containing peptide, you can actually increase it. And so you sort of see both sides of it.
They had already shown that you could actually add human thrombospondin protein and it increases it as well. So that's, I guess it's closely related. But what do you think is going on here where they could add an exogenous protein and stimulate it? Because if there's a receptor on the dinoflagellate for the TS1, I would almost say would block it, right?
No, I know it. And so, agree. And I think and they admit to it that they don't know necessarily what the mechanism is here. Because for example, you could imagine a case where maybe the dinoflagellate secretes a ligand, and then the TSR protein responds to that. And that would be consistent, so if you block basically a receptor with an antibody, then the ligand from the other species couldn't sort of stimulate that. And that, that would be, so this would be a hypothesis that the TSR protein is a pattern recognition receptor. And that basically the dinoflagellate is producing an epitope or a pattern, a protein or a peptide or some molecule that binds to that host receptor, and that sort of sets off, and that's sort of how you kind of begin that molecular conversation to say, hey, why don't you take me up into your tentacle, but don't digest me. Let's hang out and be symbionts together. But the problem, you're right, the problem with that hypothesis kind of comes to a screeching halt when you add the human TSR in the super colonizer position, right? Because now, if anything, I think what would be consistent with the pattern recognition receptor would be that would also lower the amount of colonization, because you would basically soak up the ligand from the dinoflagellate. And so I think that was...
Unless it is induced... So it could be that the TSR proteins on the cnidarian is actually inducing ligands on the dinoflagellate, right?
Indeed, yeah. And they, in fact, kind of in the end, it's a little bit kind of speculative in the discussion, but they... sort of make a case. They use the analogy of the complement system, which involves sort of a couple of levels of interactions to activate this arm of the innate immune system, and try to say that, well, maybe something like that is going on here. Like you're saying, you kind of stimulate something on sort of both sides of the interface. Yeah. But that's still, I think that's certainly a future direction.
They do look at some gene expression, right? By quantitative PCR.
Yeah, right. And so this is, let's see here, I'm just scrolling down. This is in figure 6.
Yeah.
And so they note that, so what they're comparing basically is a constant aposymbiont phase where there are no dinoflagellates present, and they sort of set that expression level for the genes they consider to 0. And then they ask what happens during inoculation or the mixing of the dinoflagellate with the cnidarian. And so for panel A, they show up RT-PCR evidence that you have right at the first 12 hours, you have a sort of a increase by a log 2 amount of this so-called semaphorin domain containing protein that also contains these TSRs or thrombus bondin domains as well. And so that one's kind of interesting. They point out that, semaphorins are known to influence actin cytoskeleton in some cases, and they try to draw at least some connection to the idea that obviously the uptake of a dinoflagellate through phagocytosis it would involve actin dynamics, and perhaps this is mediating that. There's also a couple other connections that they note. So it turns out Plasmodium, right? So these are the parasites that can cause malaria, for example. They mediate their interaction with host cells, blood cells in this case, using a semaphorin 5. And so that's sort of a more sinister case of a two species collision. It's also kind of interesting, getting back to the viruses, some of our favorites. Herpes viruses and pox viruses actually encode semaphorin proteins that they've acquired from their hosts at some point through horizontal transfer. And the idea there is semaphorins are also involved in things like axonal guidance and even immunological synapses. So this is in the adaptive immune system. The interactions that kick off adaptive immune responses are engaged or mediated through semaphorins. And so anyway, these echoes, right, of this other biology that makes them think this could be a great candidate for a symbiotic relationship as well. Yep. Yeah.
I guess part of the issue here is that there are a lot of TSR containing proteins, and I don't know exactly which ones are having the effect, right?
Exactly. And so I think, they've made a really nice, I think they've taken sort of some of the preliminary data we discussed about these TSRs being proposed as candidates. They've moved the bar forward by actually getting a little bit of a functional hold.
Yeah, for sure.
But then it's exactly what you're saying, which is, okay, now can we really, for the next steps, can they and others start to really nail down the exact genes encoding the proteins that are mediating this symbiosis? And so for that, I think what we would really hope to have are some to put into play are some genetic tools, some genetic modifications, maybe using CRISPR or other things, right? To pick out some of, maybe prioritize the TSRs, knock some out, and then ask what happens when we mix with the algae.
Yeah, I was wondering if they could do CRISPR modification in these organisms.
Yeah, it's a good question. To my knowledge, I don't know that that's been done yet. I mean, remember when we spoke about the raider ants?
Yeah, first time.
Right? And yeah, it was not a small endeavor, and it took a pretty big investment on the parts of our guests to get that one event. And so
they're trying, yeah.
Exactly. And so hopefully they'll be successful.
Nels, they say that TSR proteins can be added to the growing list of immunity genes in cnidarians that are greatly diversified compared to their counterparts in vertebrate genomes. And so this is an expanded repertoire. They say this expanded repertoire is an alternate evolutionary strategy to vertebrate adaptive immunity that would enable complex reactions to and management of their microbiomes. Can you explain that for us?
Yeah. I'm glad you brought that up. So I think two important points kind of coming out of that part of the discussion. So the first is, I think it's really important, and we've tried to highlight this a few times on TWiEVO, is that, when we're talking about cnidarians, it's sometimes tempting to think of these as sort of primitive organisms that because they don't, have conversations or host podcasts like you, like you and I do. But remember, they've had just as much time as we have to evolve. They've just kind of gone in a different direction. And so they've gone through as much or even more sort of trial and error of mutation and selection. And what they've come up with, in a sense, is a way to interact or mediate these interactions in a very sophisticated way with these algae, in a way that we took some of the same parts list or toolbox of these thrombus bondin genes that have sort of gone in another evolutionary pathway to help mediate some of the immunological reactions and even the tolerance pathways that are important for our immune system sort of not overreacting. And so I think, so that's one of the points, is just to remember that when we talk kind of from an evolutionary standpoint about, invertebrate species, they're not primitive. in any sense that they've had just as long to evolve of us. They've gone in a different direction.
Yeah.
And then the second point is, this idea of this massive expansion of protein families, and that can really, in these kind of new combinations of domains that you might just sort of sample and try out, this can be indicative of kind of this volatile evolution. And I like to kind of just speculate in cases like this that there is a little more sinister kind of underpinning to the evolution that's happened, in that you have cases where, for example, if it becomes more of an infection than a symbiotic scenario, then that might select for a new domain or a new protein that kind of keeps the other species behaved, or keeps that association more stable. And so I think that's one fun speculative point, is that maybe some of that protein diversity actually reflects kind of this natural selection, kind of on the knife's edge of being cooperative or being in an ongoing conflict.
Yeah.
And Yep.
Yeah, I think they have a lot of interesting points in their discussion where they go into the different types of proteins that these repeats are found in, and what they want to give some ideas about what they might be doing, right?
Yeah, exactly.
In terms of recognition and immune responses. One of the tracks they take is this attachment idea. In the apicomplexans, like malaria and others. These proteins are involved in attachment, right? And so they wonder if, in fact, they have a model where they are involved in attachment of the dinoflagellate to the anemone, right?
Yeah, exactly. This is figure 7, which is sort of an interesting. They're pushing this idea of these recognizing these molecular patterns somehow, right, on both sides, right? Yeah.
So they're multifunctional. They're attaching, their recognition, and then there are probably downstream reactions as well, so that the response is muted and they don't kick out the dinoflagellates, right?
Yeah, that's the idea. Or don't digest them would be another outcome. Yeah. If you get into a phagosome. Yeah, exactly. So, and it's interesting. I mean, it's kind of, I think when you read the discussion and even the introduction, it's a little bit all over the map. I mean, it's highlighting these interesting sort of this evolutionary history, but we still can't really put our thumb on what's the really most important. I mean, I think it's a good model for the information they have, but I'm sure that model will be revised maybe many times. And
well, it's not, and I'm sure this isn't the only kind of protein involved, right? There are probably others. Right. This is the one they happen to look at, and it tracked well. The results tracked well with playing a role, but I'll bet there are others.
Oh yeah. Yep. Yeah. This is, I mean, you kind of pick this kind of science you do.
Yeah.
And so one of the things like I admire about this work is that it's kind of in its infancy, in terms of having the tools to really address at a mechanistic level how these things play out. And so for some people though, they would look at it and it'd be really frustrating because it's like, well, come on, you don't really know what's happening.
Yeah.
And on the other side, you could say, wow, this is really exciting. We don't really know what's happening here. And so you kind of have you can kind of modulate your scientific taste in a sense based on the kind of work that you like.
Wouldn't it be interesting to do a transcriptional analysis? You take the anemone on its own, you'd look at the RNAs that are produced, and then throw in some dinoflagellates and make RNA and see both dinoflagellate and anemone, what they're making?
Agree. And so I think this paper really focuses on the cnidarian side of the interface, right? Yeah. And in a sense that, kind of the host side, which maybe we have some sympathy for. But you're exactly right, that the dinoflagellates, right? That if the model holds water, what are the molecular patterns? And are those, I think one definitely useful way of going forward is to ask, okay, well, are they upregulate upon that interaction? Are they upregulating genes encoding potentially proteins that are kind of the other half of that conversation in a sense?
Yeah, so, they could modulate the anemone so it doesn't digest them, right, in some way.
There you go. And I think having that sort of data set could be a important way forward back on the cnidarian side, right? Yeah.
And how cool would it be if there were some dinoflagellates which are kind of closely related but get digested?
Exactly. That would be, the other side of the coin for thinking about it.
Compare them, right?
Yeah, great idea.
Now, Nels, in a broader sense, these you made the point before they're not really primitive just because they can't podcast. Right?
Mm-hmm. Right.
So do we?
Which is the highest form of life is podcasting, right?
Oh, it certainly should be the bar. It should be the bar of higher intelligence, right?
There we go.
What do we think that these anemones and these dinoflagellates look pretty much like organisms that were around millions and millions of years ago?
Yeah, right. And so I'm glad you're bringing that up, because that's sort of, I think, where the confusion can arise, is that when we talk about these guys as primitive organisms, what we are actually meaning is that we're inferring that this might be closer to a more primitive form that our ancestors sort of went through.
Right.
And then continued on. The thing to remember is, well, so first of all, we shared a last common ancestor with the cnidarians. So at that point, we looked identical. And then, and it might just be mostly from a morphological standpoint that some of these cnidarians maybe look a little bit closer to our last common ancestor than we do. And that's where some of the confusion can arise, because it's sort of that primitive appearance morphologically versus the fact that if we sample a cnidarian or if we sample a primate today, we have had just as long to live as we've diverged from that last common ancestor.
Yeah. So they may be very different, right?
Indeed. Yep. That's right.
What? So that begs the question, and I know the answer, but I'm sure our listeners would like to hear that. But when you have this evolution to something like a podcasting primate, why do we still have anemones around?
So in there, I think, one of the simplest arguments is that we've kind of a lot of species, as they've diverged, have sort of filled different niches. Right. And some of which are very similar to what was around a long time ago.
Right.
And so at least from a morphological standpoint, or even in some cases a biochemical standpoint, you haven't had to, you don't see these sort of dramatic adaptations, because the environment, relatively speaking, not completely, but relatively speaking, has been very much the same. Whereas some of the, not to put too much of an anthropomorphic spin on it, but some of the more adventurous species, so to speak, who've kind of ventured out to the Arctic polar caps or to, high elevations, have sort of faced the selective pressure that comes with adapting to a harsh environment. Yeah.
I guess it's all about the niche, right? Because in the end, the niche where the cnidarians live, and there are many, but they're really well adapted to that, right? And we're not. We're not adapted to that aquatic niche at all. And so there's room for what looks like on the surface a relatively simple organism. There's no point in it getting more complicated or bigger. It wouldn't work well in a marine environment, right?
Yeah, that's right. And it all depends on kind of, it's all sort of what your point of view is. So if you thought, so you and I might make an argument that podcasting is the pinnacle of evolution. A cnidarian might make the argument that being able to accommodate a dinoflagellate that gets into your cells and then just makes energy for you, that's the pinnacle of evolution. And by the way, wouldn't that be pretty cool if we came across some algae, could just rub it on our arms and we wouldn't have to eat a meal for a week? That would be a pretty cool trick. I don't know how to do that, and our evolutionary ancestors never figured it out. Yeah. And so we're really bad at that. Yeah.
I want to anticipate a listener complaining about my comment that these little anemones are well adapted, and I said they don't want to get any bigger or more complicated. Of course, there are more complicated organisms in the marine environments, but it's a different niche, right? And these little guys are living in a certain place, and what they are works really well for them.
Yeah. Although I'm glad you're bringing up this point, Vincent, because what has worked well historically is starting to break down, at least for some of these species. And I'm thinking about the coral reefs in particular, and this idea of bleaching of corals, where they lose their dinoflagellates. That's exactly what's going on here. And so as oceans are becoming more acidic, as the temperatures are going up, it's really, I think, breathtaking and horrific to look at what's happening on the Great Barrier Reef right now in terms of how that ecosystem or that habitat is changing dramatically, and the potentially disastrous consequences that's going to have for the reef. Yeah,
I've seen some talks where they show photographs of some of these bleached reefs, and it's just devoid of fish, because a lot of fish live in these reefs when they're healthy, right?
That's right.
They're gone, and it's really scary what's going to happen there.
Agree. We're doing this massive uncontrolled experiment on the resiliency of these reefs, which so many other species depend on, without really knowing what's going to happen. It's really scary.
I'm going to tell you about the sponsor of this episode, Blue Apron.
Yeah.
Fresh, high-quality ingredients make a real difference, so it's important to know where your food comes from. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. They set the highest quality standards for their community of over 150 local farms, fisheries, and ranchers all across the U.S. So there you go. You know exactly where your food is coming from. They have this community of suppliers, and they make sure it's good. For less than $10 per person per meal, Blue Apron delivers seasonal recipes along with just the right amount of the ingredients you need to make delicious home-cooked meals, and you can do it in 40 minutes or less. And they ship you exactly what you need for a recipe, so they're reducing waste. Plus, you can't overeat. They only give you so much. Of course, I guess you could make 10 recipes, and then you could overeat. But
I was just thinking I always find a way to overeat under most circumstances.
You just make only one. Their mission is to make incredible home cooking accessible to everyone. They deliver to 99% of the continental U.S. There's no weekly commitment, so you only get deliveries when you want them. I've tried it. It's great. It's easy. Yeah. I like cooking, but I don't like shopping. I hate shopping for all the stuff that I need, and then I inevitably get home and I've forgotten something.
I hear you.
So, Blue Apron, they give you everything. If you need a teaspoon of soy sauce, it comes in a little bottle with just a teaspoon in it. It's really great. And they're fun to do. It's quick and relatively inexpensive. Have you tried this yet, Nels?
Well, I keep meaning to, Vincent. And finally, I know, but finally, after ASV in a couple of weeks, my travel schedule is really simmering down. And so I'm going to take a look at Blue Apron.
Yeah, you'll like it. Here's some new recipes that are coming up. You can choose yourself, by the way, or you can let them surprise you. They can send you random stuff. Some upcoming recipes include warm smoked trout and asparagus salad with fingerling potatoes and garlic crouton.
Ooh, that sounds nice.
Spiced zucchini enchiladas with creamy lime and tomato rice. Wow, that's good. Yeah. Elote-style vegetable tostadas with summer squash, poblano peppers, and cilantro rice, and peach honey-glazed chicken with mashed sweet potatoes, collard greens, and Thai basil. As you can see, they have fish, they have meat, they have vegetarian options. You can customize your recipes every week based on your dietary preferences, and they have a freshness guarantee. They promise that every ingredient arrives ready to cook, or they'll make it right. Check out this week's menu and get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiE, TWiE. That's blueapron.com/TWiE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron. So don't wait. Blue Apron, a better way to cook. Now that I'm sufficiently hungry.
That's right.
Well, here it's almost 5:30, so
right.
Can head on home in a bit and just cook it up.
Good idea.
Let's read some emails. Nels, we've had an accumulation of a bunch because we have had a few guests. We haven't read any.
Yeah, good idea.
First one is from Rafik, who writes, "Hi, guys. I'm a fan. I like Nels' work in particular ever since the Malik Lab. I remember meeting you at a Gordon Research Conference in Easton a few years ago." Where's Easton?
Massachusetts, suburb of Boston.
Yeah, they have Gordon Research conferences there.
Yeah. Say again?
They have Gordon conferences there?
They do, yeah. I'm forgetting the name of the university. It's a small, kind of a liberal arts school.
Neat.
And in fact, that was exactly 2 years ago. So that conference just started today. Molecular Mechanisms of Evolution. A couple of postdocs from my crew are out there representing the Elde lab.
He continues, "I did not know Vincent before. I must shamefully admit." Yes, you're ashamed.
There you go.
It depends what field you're in, but I'm amazed by the scientific and communication work. I've started spreading this podcast among my EVO friends, but also non-scientists, which has been a gateway to the other TWiE podcasts. I was mind blown by the stuff you ant stuff.
Ant stuff, yeah.
You presented in the last one. Yeah, that was mind blowing.
Yeah, agree.
I work on the evolution of molecular innovation, and with my former advisor, Diethard Tautz, MPI for Evolutionary Biology in Germany. We're having a paper out next week in Nature TWiEVO on the effects of random sequences in E. coli cells and the implications of this in the field of de novo gene birth. It also seems to be of broader relevance for pharmaceutical and biotechnology developments. Are you aware of Dieter Tautz?
I am. So I met him at that meeting as well 2 years ago.
Yeah.
And I remember talking to Rafik about this really interesting work. Yeah.
We basically transformed a population of cells with random sequences and had them compete while expressing them to show which ones had good, bad, or neutral effects for their growth. It's been my favorite story so far, and I'm really proud to finally put it out there. Curiously, with Joanna Maisel from University of Arizona, we're having another paper in the same journal about how all of the possible new genes that can be born, only those matching certain conditions seem to ever make it. This is totally a coincidence since those were completely independent projects that started around different times. The main point of that paper is that de novo gene birth does not follow a smooth transition from non-genes to genes, but it's more like a quantum leap based on disorder predictions. I'd like to know if this is interesting enough to you guys to feature at some point. They're coming out this Wednesday, but I can provide you with copies ahead of publications. Thanks for spreading good science and keep up the good work. Big up from New York.
No, there we go.
Rafik.
In your backyard here.
Yeah, he's right here. He's in the Department of Biochemistry and Molecular Biophysics right here at the Columbia University Medical Center. And, he said, I must shamefully admit that he didn't know of me. Well, I must shamefully admit that I didn't even know you were here.
Well, that's great. This podcast can bring us together.
Yes. Well, since you're here, we should have him over, right?
Oh, definitely.
Sometime. Talk about
definitely.
This stuff.
No, that would be really fun. And actually it kind of echoes a little bit on what we were just talking about today. So what I'm not going to remember all the details, but this idea of de novo gene birth today, we're talking about kind of scrambling or mixing and matching these domains.
Yeah.
Which is, but you already have these domains kind of pre-made and it's like the blue apron of protein evolution. That's right. You've got you're just enough of this, just enough of that, and you combine it. What Rafik is talking about is, like nucleic acid sequence, random sequences, basically now clicking from non-coding to coding and actually taking on a function, which is, even a more kind of dramatic, in a sense, evolutionary step in terms of gene coming online or the origin of a new gene. Really interesting work. Yep.
I like this idea that, gene birth is not a smooth transition. It's a quantum leap, right?
Yeah, yeah.
Because when we think of evolution, we think of things that are really slow and one base change at a time, but it doesn't have to be that way.
Mm-hmm.
And
exactly.
There's no trajectory, right? It's just for the moment. So it makes sense that it would be a quantum thing.
Mm-hmm. Yeah, and then here you get into these issues of disorder, right? As proteins fold up, what's tolerated and not tolerated, and how you sort of go from something that's not really workable to something that could work, and then more so than that actually gains a function and then is kept around. It's a really fascinating emerging field in evolutionary genetics.
You take the next one, Nels?
Yeah, sounds good. So this is from Anthony. And Anthony writes, in the audio on the protozoan Toxoplasma available here, and there's a link to a microbe blog, an experiment is mentioned where an attempt was made to determine if there was a correlation between the parasite and people's perception of cat urine. There was one, but only for men. So as it stands now, there is no established connection between Toxoplasma and what were described as crazy cat ladies. We've talked a little bit about toxo and this idea of controlling host behavior, making mice and rats unafraid of cats. And then this would be sort of a loose extension on this hypothesis that, many humans are infected with toxo, and whether this could sort of, underlie an affinity for cats. And this would be, if you really push the hypothesis, the crazy cat lady idea. Tangentially, though, audio can still be accessed at the link. The formatting in the page is mangled. This is at that blog he's describing. I've tried to bring that to the attention of ASM, but the page still looks strange. Perhaps you might know who to mention it to. It's believed that toxo affects mice so that it's easier for cats to catch them. It's believed that Bilascaris, I'm totally mispronouncing it.
Bilascaris.
Thank you, Vincent. Yep, another parasite. You've had enough time with Dixon to polish up on some of these. By killing small animals, this roundworm makes it easier for raccoons to catch them. By the way, in the two preceding sentences, them can be understood as either the prey or the parasite. So then Anthony gets to a question. Might the various mosquito-borne ills through virulence make it more difficult for people to prevent the insects from biting? In Alice in Wonderland, one side of the caterpillar's mushroom enlarged and the other side shrank. Might it be that some cases of the caterpillar's mushroom pattern in the triangular interactions between two hosts in a parasite? In one host, the predator, virulence diminishes. In the other host, the prey, virulence increases, or there's some other effect that makes it easier for the predator. In the prey, there'll be the red queen. And will the predator host tend to change to accommodate, run to instead of away from the parasite? I might be wrong about the cat urine experiment being mentioned on the ASM audio. It is featured in this YouTube video. And then he has a link to the YouTube video as well, which we can put up. Wow. So we covered quite a bit of ground here, Anthony, including into Lewis Carroll's Alice in Wonderland and extending. This is great. I mean, so the Red Queen hypothesis, which we've talked about some on the show, is sort of borrowing from Lewis Carroll's character, the Red Queen. And she says to Alice through the book, Through the Looking Glass, takes all the running you can do to stay in the same place, which is that reference to the Red Queen. And here, Anthony is proposing, I think, an interesting Alice in Wonderland reference to the caterpillar's mushroom, enlarging in one side and shrinking on the other. Whether that's really great. Whether the predator, or depending on the interaction, that the predator could actually, instead of running to stay in the same place or running away, in a sense, would actually sort of team up with a parasite. Whereas if you're the prey, that you would be susceptible to that. I think that's probably actually right. So if we think about, I'm just trying to think right now about, if we think about some insect, not just the caterpillar's mushroom, but caterpillars themselves, actually, literally, where parasitic wasps will deploy viruses when they deposit their larvae. Mm-hmm. And that now you have sort of a virus being harnessed or domesticated to help turn off the immune system of the caterpillar to allow the wasp larva to develop. And so maybe that's a case that fits that. I haven't thought too much about it, but I think basically what you can say is that anytime you start to collide multiple genomes again, in this case, predators and preys, or parasites and hosts, then the genetics outcomes become complex in a hurry, and you can get all of these interesting and complicated outcomes.
So he has this question about mosquitoes. Could mosquito-borne ills through virulence make it more difficult for people to prevent the insects from biting? Well, of course, can't prevent you from letting them bite you before they've bitten you, because then they can't modify your behavior, right?
That's right.
So, yeah. But what they can do when mosquitoes bite, we know they inject anesthetics so Correct. That you don't slap them right away. You don't feel the bite, right. Basically. So they have evolved to do that, I presume. I mean, there were mosquitoes that kept getting killed, and then one just popped up with an anesthetic, and well, maybe it's not as simple as that, and that one survived, and all its offspring survived.
Yeah, well, then you can get in, I don't know if anyone's looked at this, but you can get into these ideas of group selection. So if that anesthetic is, depending on how far that disperses, mm-hmm. that could mean that then, so there's sort of the pioneer bite, but then as other mosquitoes show up, they might have an easier time.
Could be. Yeah.
And if that were true, then, so the fitness advantage you might postulate under a scenario like that would be that, if your close relatives are gaining more calories mm-hmm. from these hosts, mm-hmm. then all of a sudden something could happen. But those are really tricky hypotheses to test.
Would that be like public goods?
Yeah, it could be, or a tragedy of the commons. If you got stung by 1 billion mosquitoes at the same time, pretty soon you'll lose your host meal, right? That might be tragedy of the commons.
You can imagine a lot of these sorts of scenarios, but in the end, they're hard to test, right? And so they remain kind of speculative, but they're interesting to think about, right?
Yeah, that's right. And then so that's where, as more genetic tools come online, or we have more access to genomes, where you can start to get a little bit of a foothold, maybe we'll get better and better
maybe
digging in. Yeah.
Next one is from Alexandra. Dear Nels and Vincent, first of all, I must write that the two of you have planted an evolution seed in my mind. I am an undergraduate in immunology, but came from chemistry background and have never took a course on evolution. I've been studying biology for 3 years now, but have completely failed to realize how fascinating evolution is. Several factors contributed to my recent awakening and realization that evolutionary biology is amazing, but TWiEVO has undoubtedly played a major role. I was completely sold after the episode with Dr. Hopi Hoekstra.
Yes, me too.
Now to my question, a bit of background. I'm a final year undergraduate of immunology. I've been accepted to a graduate program in immunology, and I'm currently thinking about what I would like to study. I will be able to do 3 rotations, and so I'm trying to identify faculty that are doing interesting work. Before, I haven't thought much about the evolutionary processes behind immunological phenomena, but as I seem to have caught an evolution bug recently, I would like to learn a bit more about this. My question to you is, do you know of any professors at Harvard that are interested in evolutionary immunology or are currently doing research that is somewhat related to how immunological mechanisms evolved? As I have said, I'm very new to the field, and the short descriptions in previous papers often do not reflect what a given PI is currently working on. So it's a bit hard to identify faculty who would be interested in such problems. For instance, one question that comes to mind, especially as interest in neuroimmunology has been on the rise recently, is why and how neuro and immune systems co-evolved, which seems to be the case. There is some work done on this, but I haven't seen many papers. I know I have to do much more reading as I don't have more specific questions in mind yet. However, I thought that maybe 2 of you would know of scientists in Boston who are doing some evolutionary immunology work. In any case, again, I love the show. Vincent, you are a veteran in science podcasting, so it goes without saying that you are amazing at it. But Nels, I guess during one of the first episodes, Vincent mentioned that when he met you, he knew you would be great at this, and he was 100% spot on. Thanks a lot for your efforts, though I wish it was actually this week rather than this month. Not complaining, though. Best. Alexander's at the University of Edinburgh.
That's great.
Yeah,
thanks a million.
I'm not sure I'm amazing, but I can spot talent when I see it.
Uh-oh. No, thanks so much, Alexander. That's really a kind note.
Sounds like she's going to Harvard, right?
Yeah, or aspiring to.
Well, she said I'm going to be an undergrad, and I've been accepted to a graduate program.
Oh, good for her.
So do you know anybody at Harvard doing this?
Great question. I mean, so Hopi would be she's not doing a ton of directly immune related stuff, but she knows everyone in the neighborhood.
Should email her. Yeah.
Yeah, exactly. And I think, what I would suggest, and I'm just thinking back to my own, when I was looking to sort of break into more evolutionary biology from cell biology, I spent a lot of time just sort of sifting through lab websites and talking to people and getting their ideas. And I came across this guy, Harmeet Mullik at the Fred Hutch, who was thinking about kind of combining evolutionary ideas with some experimental approaches. And we really hit it off. And, the rest is history, as they say. But yeah, so I think it's actually really worth it to spend some time asking a lot of people, and especially people in the neighborhood who might have a ground level view of what's happening or who's thinking about some of these ideas. I will say that, almost anyone right now in virology at least has the idea of evolution in the back of their mind. And one of the fun things thinking about as you're sort of progressing in immunology, if you're bringing some new evolutionary questions to the table, depending on what they are, you might really spark the interest of a potential mentor to sort of pick up on even some new ideas and sort of bring this together in the systems that they're working on. And so I wouldn't even sort of limit myself to just, people who are already really thinking about evolution in sort of really rigorous ways. So the one lab that does come to mind, Vincent, is, have you run across this guy, David Haig, Haig?
No, have not.
Yeah. So he does a lot of work on the evolution of imprinting and sort of maternal versus paternal contributions to offspring, and how this can actually look like a conflict. How you invest in your offspring, if you're a mother or a father, can be two very different things. And actually, this has some real implications, even from a neuronal or standpoint. And so along with David Haig, there's Catherine Dulock's lab. She's more of a straight neuroscience lab. But some of that thinking has emerged, or sort of merged together, both the evolution and the neurobiology. And in fact, there's a guy here at Utah, Chris Gregg, who came out of Catherine's lab, and he's thinking about a lot of these questions as well.
Cool.
And so I think there's tons of possibilities. And the deeper you dig, either on the immunological side or the neurological side, I think you'll run into really good opportunities. So Mike writes, "Hi there. I've been going back and listening to the archived episodes since I found this podcast and really enjoying some of the episodes. The question that was asked in number 9 by Alex about genes which modulate the distribution of phenotypes, sort of a metagene, reminded me of a great paper about exactly that in Bacterial Motility, which I've attached. Feel free to forward it to Alex." So in the pub, he has a PubMed link here to a paper from 2016 called "Non-Genetic Diversity Modulates Population Performance." And indeed, I think this looks pretty interesting. Have you taken a peek at this?
Yeah, it's pretty cool. They have a microfluidic device to measure the swimming of E. coli.
Yeah, that's great.
They give them a gradient, an attractant gradient, and they can look at how the structure of the population emerges, from initial populations that they put together. And so that is, they can make a phenotype to performance map and show this, that it's cool. As he says, this is answering the question about genes that modify the distribution of phenotypes, and that's what they're doing in this paper.
Yeah, so they say by manipulating the expression of key chemotaxis proteins, we establish a causal relationship between protein expression, non-genetic diversity, and performance that was predicted in their sort of theoretical models. Yeah. It sounds like a cool system.
Thanks for that, Mike. And maybe Alex is still listening, and he'll find it.
Yeah, I hope so.
Next one is from Michael. "Hello, Nels and Vincent. I thought I would share a video that Julie Theriot made about 20 years ago when she talks about her decision to go into science and explore the wonder and awe of it all. I found it a very inspirational, especially the part of how slime molds can go from a unicellular organism to a multicellular-like worm. I was familiar with her work and that of other great scientists, including your guest, Hopi Hoekstra, through iBio seminars, and highly recommend that resource. Also, keep up the great podcast, Mike in Oregon." And Julie Theriot, good stuff.
Yeah, agree. One of my science heroes. She's great, really great.
I heard her talk at ASM in New Orleans.
Just a few weeks ago, a couple weeks ago.
She was in the initial session. Really cool. She was great.
Yeah,
and
she's spectacular.
This is funny because this is a young Julie here interviewed, and it's like 20 years ago.
That's cool.
Yeah. Anyway, she's a professor at Stanford, and it's funny. The session she was in has another investigator who is the basis for my pick of the week.
Ah, cool. We'll get to that in a second. But yeah, so slime molds, right? This came up even, dictyostelium. This came up even in our last TWiEVO with Jonathan Weiner. He was telling us about some of his first writing was on slime molds, and that kind of got him inspired. So these things, these are pretty charismatic critters.
All right, take that last one.
Okay, so this one's from Matt. He says, greetings from Jena, Germany. I'm sure you hear this sentiment a lot, but I've really been enjoying the TWiEVO podcast.
We don't hear it enough, right, Nels?
Yeah, keep it coming. This is having a blast here reading these letters. It's really nice. In the most recent episode with Jonathan Weiner, you all touched on the idea that scientists don't spend enough time just thinking or chatting science over dinner. Perhaps it's not a fair comparison, but I think your podcast is a way for those of us stuck in the lab to get a bit of a dose of that conversation. I do agree that we all need to spend a bit more time thinking outside the laboratory box, though. Anyway, just wanted to say thanks for an excellent podcast. Best, Matt. Thank you, Matt. Really glad to hear that you're enjoying TWiEVO. And I have to say, I know Matt. So he was here, got his PhD at the University of Utah, and is now doing a postdoc over in Germany. It's good to hear from him, and it sounds like he's doing really well.
Well, I think that was the original idea behind these podcasts, is that we have conversations about science, kind of casual, and that people could listen in, right?
Yeah, I agree. And I'm just, thrilled that people are kind of hanging out in the lab and listening to us drone on about some of these ideas, and that it's actually something that's a positive conversation. And the more folks who want to join us in the conversation, the better. And so thanks for all these letters, and thanks for your support. And it's really, really great.
You bet. Yep. All right, let's do some picks, Nels.
Yeah, so someone alerted me to what I think is kind of an interesting exercise in science communication. And this is a video. It's called CRISPR 5 Ways. And so the idea here is to explain a scientific concept to 5 different audiences. So you start by explaining whatever the science is to a 7-year-old. Then you do it to a 14-year-old, then a college student, then a graduate student, and then finally to sort of an expert in the field. And it's a really cool idea. So sort of science 5 ways. In this case, they're specifically talking about CRISPR. So this is a fellow named Neville Sanjana. He's an assistant professor at NYU and at the New York Genome Center. The video, and we'll put the link up, was originally produced by Wired magazine. And then it got picked up by a blog at NPR called Cosmos and Culture. This is curated by Tanya Lombroso, who's a professor of psychology at Berkeley. And then now we're echoing it for the 3rd time, I guess, at least, over here on TWiEVO. But so Neville sits down in this video and first talks with a 7-year-old, a 14-year-old, a college student, a graduate student, and an expert. And it's really interesting to see how those, to kind of in rapid succession, see how the conversation and the concepts sort of emerge at different levels of expertise and interest. This might be a good exercise for other science communicators, myself included, to maybe think about tinkering with and seeing how useful that might be.
Yeah, these sorts of things are great exercises, and when you see them, they're very entertaining. Yeah, you can learn a lot from them. Yep, for sure.
Yeah,
that's pretty.
It's also
cool.
interesting. Agree. It's also interesting how it goes from very much sort of a one-sided discussion to a conversation as you kind of continue, and to think about, how could I maybe have a conversation in each setting, possibly, and sort of as you're adjusting your sales pitch, so to speak. Yeah, really interesting.
I wonder what is our pitch here on TWiEVO aimed at? What do you think?
Yeah, good question. And folks have asked me that. I mean, the way I've been thinking about it, and I don't know if it's true or not, is that we're sort of aiming for, maybe an advanced college student, or a really interested college student, or grad student who's starting out, or someone who is in a different field but is sort of evolution curious, as we heard from one of our letters, Alexandra's letter before. But the truth is, of course, I think both of us would love to capture kind of as broad of a swath or a demographic as possible, both scientists and non-scientists, at all levels. And so it's really a challenge, right? Because there's so much sort of language and certain concepts that we almost take for granted without thinking about.
Well, that's what this video points out, that you have to address people differently, right? So I'm not sure we could capture a 7-year-old with TWiEVO at the moment. Maybe some 14-year-olds, but pretty rare.
Yeah,
I would love to have high school students listen. We have a few on TWiV. They write in now and then, so I know they're listening. But we talk about complicated things, or not necessarily complicated, but we use jargon a lot, and don't take the time to explain it all. And I guess we could shorten it up and cover a lot less and just explain it a lot better. And these are things that we can always modify and fix as we go on. This is a podcast, and it evolves just like everything else.
There you go. That's right. Yeah. No. And the more feedback we can get from folks who might say, this worked or this didn't work, or how we're pitching it. I think at least for me, the bigger picture is what one of our major goals is to lift up interesting new work that sort of bridges the fields of evolution and experimental biology, and then see kind of where this might go, and to try to inspire others to kind of think broadly and about some of this stuff. And so that kind of, triangulates us to some degree, perhaps.
Yeah.
How about you, Vincent? What's your pick of the week? You previewed it a little bit earlier.
So my pick is a book by Mark Lane. I had not known Mark Lane before I went to ASM Microbe in New Orleans 10 days or so ago. Do you know Mark Lane at all?
No, I don't.
Oh, you're in for a great pleasure here. So he gave a talk at the opening session in which Julie Theriault also spoke. Now, Nick was the last speaker, and he was just awesome. And he's a professor he's a professor of evolutionary biochemistry at University College London. And what I didn't realize is he's published 4 books, which have all sold really well. So I bought one of them and I read it. It's called The Vital Question: Why is Life the Way It Is? His other books are called Life Ascending, 10 Great Inventions of Evolution, Oxygen, the Molecule That Made the World, Power, Sex, Suicide, Mitochondria, and the Meaning of Life.
Oh, yeah. Actually, I have come across, I haven't read it. I'm a little embarrassed to admit I haven't read any of these books, but I have heard really great things about, I
mean, he gave a great talk, which is all about, his thing is how really it was energy that drove the evolution of eukaryotes with their multicellular structures and really more complicated structures, hate to use the word complicated now, but, I think you understand what we're saying. Bacteria are single cells pretty much, even though they live in communities now we understand, but they are not multicellular organisms. And he thinks this has all been limited by energy, and that's what his theme is in all of his books. Now, The Vital Question focuses on that. He talks a lot about how we get energy and, it's all about electrons and so forth. And, it starts with the idea that, the mitochondria was really important to development of eukaryotes. And there's a theory that is kind of contrary to Lynn Margulis. Right?
Yes, exactly. That's exactly right. Yep.
She said, eukaryotes captured bacteria and they became mitochondria. There's another theory that says that it's actually an archaea that captured a mitochondria, and he puts forth the evidence for that, which is quite compelling, based on lots and lots of genome analysis. It was archaea that captured bacteria, which eventually became a mitochondria and then became eukaryotes. So there's a new tree of life that he shows.
Yep. Where
it's not 3 branches of life, archaea, bacteria, and eukaryotes, but they're all intertwined because they contributed to each other. Right. So I think this is the thinking of Bill Russell, who he particularly likes. So anyway, this book is all about understanding why he thinks energy drove the enormous accelerated evolution of eukaryotes. And it's just a wonderful book. It's putting evolution starting at 4 billion years ago in history to the present. I love that sort of thing where he takes all the evidence we have at the moment and puts it in a historical context of what happened. And he's lots of, he's got now protein structures of energy producing proteins and how they're similar across, domains of life. It's just beautifully written. And he talks to you and he says, here's what I'm thinking. This is what I think. And let me explain why this is really important now. And we have to go over this and this and this. It was really, really beautiful writing. I love it. I just love this kind of book where you take a big picture, and I so admire people who can do this, I wish I could do it for my field.
Totally agree. It's a great thing to aspire to. I mean, we're coming at it from another direction, right? From beginning our career as scientists, and then sort of converging or approaching some of the space that some of the writers or communicators started on sort of the other side. Yeah, and actually, the more you're talking about Nick Lane, I mean, the more this is jogging my memory. And so if you look at his book from 10 years ago on mitochondria, the title is Power, Sex, Suicide, which I think hints at that, when we think about the symbiotic relationships that we've been even talking about today, that they're not necessarily as kind of well-behaved or stable sometimes as you think. I mean, there's and he's really good at sort of just exactly what you said, highlighting.
So what I like, he's a scientist, right? And he's doing research like we are, and he writes these great books. Now, Jonathan Weiner, great writer. There's nothing like him. You will not find prose in Nick Lane's book like Jonathan Weiner at all. Okay, Jonathan Weiner is a professional author who happens to write about science. And I told him that, and he was okay with it. But here, this is Nick Lane, who is a scientist who happens to write, and he actually does a really good job writing. All right. Which I think is great, because I want scientists to be celebrities. I want them to be up there. We've had a few like Carl Sagan and Neil deGrasse Tyson and so forth, but we have to have more life scientists who get up there and who are a draw and so forth. And I think he almost fits the bill. I mean, he's not a Richard Dawkins, but he could certainly be it. He's great. Anyway, his talk was great, and I immediately downloaded his book and read it. Nels, you'll love it. I know you don't have a lot of time, but you ought to check it out.
I appreciate it. I'll pick it up. And maybe we could even, reach out to him at some point and see if we can convince him to come on to TWiEVO.
Yeah, I think he's got to come on for sure, because it's all about evolution. This is the perfect podcast. I'm sure he would do it. We just have to hook up. And so maybe he's here in the U.S. at some point and we can grab him.
Yeah, good idea.
But we'll keep I just think it's wonderful. Great stuff.
Yeah, cool. Great pick.
All right, that's TWiEVO 20. You can find it at Apple Podcasts, microbe.tv/TWiEVO. If you like what we do, please think about supporting our work with a contribution. You go to microbe.tv/contribute. For example, we have a couple ways that you can do that. We have a Patreon account where you could give us $1 a month. And that's not just for TWiEVO, but for all the shows that we produce. That's a pretty good deal for like 4, 5, 6, maybe 7, 8 shows a month, and helps us to expand and do more. We want to make more shows and influence people in different ways, so that would really help. And I think you ought to subscribe to the podcast. You can do that on your phone or your tablet. There are lots of apps that you can use to subscribe to the podcast. They will automatically download an episode, and it's important to us to have lots of downloads, because that's enabled us to raise more money and help expand the program. So please do that. Of course, if you have questions and comments, TWiEVO, TWiEVO at microbe.tv. Nels Elde is at cellvolution.org, and you can find him on Twitter, Earlybird. Thanks, Nels.
Hey, thank you, Vincent. Great to be with you again.
Always good chatting.
Yeah, good to catch up.
I'm Vincent Racaniello. You can find me at virology.ws. I want to thank the sponsor of this episode, Blue Apron. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.